All right, so hello everyone. Welcome to today's Museum seminar. So we have a treat today. It's really my pleasure to welcome today's speaker, Dr. Marc Jenkins from Minnesota. I think I really do not have to give too much introduction because I think a lot of us already know his work or learn his work from textbook. Marc actually was born in Minnesota and then got his bachelor degree in microbiology from University of Minnesota. And then he continued his PhD study with Steve Miller at Northwestern University, which after he completed his PhD training, Marc actually did his postdoc training with Ronald Schwarzer at the NIH, and joined the faculty of University of Minnesota in 1988. So since then, he stayed in University of Minnesota to today, all the way from assistant professor to full professor. And then he also currently is the director of University of Minnesota Center for Immunology. As I just mentioned, Marc makes so many significant contributions in basic immunology. His lab developed a lot of novel technologies and tools which enable not only his lab, but also the entire community to study co-stimulating molecule. One of the examples is CD28, I think a lot of us also know, and try to understand and how and where the helper T cells respond to antigen in the body. And then just a few examples what so far Marc and his lab has done for the immunology community. He actually discovered the T cell secondary signal, and then also he discovered about the anatomy of CD4 T cell response, and also epitope-specific polyclonal CD4 T cell repertoire. And also his lab also discovered the polyclonal antigen-specific B cell responses. He and his lab really put a lot of effort to investigate CD4 T cell and B cell activation in vivo by also a lot of direct tracking techniques. And then the general research goal or program in his lab is try to understand lymphocyte activation, which can be used to improve vaccination and prevent autoimmunity. So with all different achievements, Marc is awarded as a Pure Scholar, and he's also the recipient of American Association of Immunologists. He's also the used to be the president of AAI, and then he is also the member of National Academy of Sciences. But I just still want to, before I let Marc get into his talk, I just want to really emphasize Marc actually has really wonderful mentorship. Both his training has achieved significant or contribute significantly in both academia and the industry. If anyone is looking for advice about how to become a mentor for the team or for the lab, I think maybe I would recommend you can just go online to check Marc's interview in AI about his career path, which I'm sure will definitely give you some idea. So without further ado, Marc, we are very excited to your talk. Please go ahead.
Thank you, Chuan. It's great to be here, and thank you all for coming tonight to hear about my research. And I've spent my whole career working on helper T cells, and I'm going to share one aspect of the work of my group related to the title shown on the screen. So I want to start by just giving you a preview of what I'm going to say to kind of pique your interest and keep you awake here at this late hour. So I'm going to talk about CD4 T cells, but in particular, I'm going to talk about T helper 1 cells. So the gamma interferon producing a subset of helper T cells. One of the, we've known about these cells for a long time, and I want to try to convince you tonight that although these cells are well studied, they're not boring. We haven't learned everything about Th1 cells. There are still, a lot of things to learn. There are There's more diversity within this larger subset of cells, and they do a lot of interesting things functionally and are in different locations in the body. So I'll say a little bit more about those things tonight. But I really want to focus on this concept that helper Th1 cells and basically all non-TFH cells form from a core circuit that simultaneously produces T helper cells for cell and humoral immunity in many infections. And I would go as far as saying in all infections, whether they are involved type 1, type 2, or type 3 immunity. And so I'm going to try to make this point and lump a lot of stuff together in a field that likes to split. And then finally, I'll make the point that, a big reason helper Th1 cells aren't boring is that if you don't have them, then there is really a 100% chance that you're going to die from an opportunistic infection, and a very specific kind of opportunistic infection. And so I'll tell you a little bit about that and raise some issues there about why we don't have effective vaccines for these kinds of infections, and how that's a challenge really for the future. So, as I mentioned, I'm going to talk tonight about CD4 T cells. These, of course, are cells of the adaptive immune system that use T cell receptors to recognize peptides found in MHC2 molecules. And those peptide MHC complexes form when proteins are taken up by phagocytes or B cells into intracellular vesicles where cathepsins cleave the protein into peptides, some of which will have anchor residues that can bind to the MHC2 molecules of these cells. Those complexes are then tracked to the cell surface, where if you're lucky, you'll have a T cell in your repertoire that can recognize that complex. TCR engagement then transduces a signal into this T cell, causing it to become activated. And that activation program, to achieve its full potential, requires other signals, including so-called signal 2 from CD28, when it interacts with its ligand CD80 and CD86 on antigen-presenting cells, and then cytokines from the innate immune system that will then drive these T cells to proliferate and then form a large population of effector cells that can effect the elimination of the pathogen from the body, at least if you're lucky. And then some of those effector cells will survive this primary immune response to become memory cells that can participate in future immune reactions to this particular microbe that contains this peptide, and then provide protective immunity. And so we have many really important theories in which CD4 T cells are central. But I'm going to argue that we really haven't integrated those theories very well, leaving, I think, a sense of confusion. And it's been confusing to me working on these T cells for 35 years as a PI. And so they're in the middle of one of the really oldest theories in adaptive immunity. And that is that there are two kinds of adaptive immunity: humoral immunity based on antibodies, and then cell-mediated immunity based on T cells. And so the early forms of this idea, of course, go all the way back to Metchnikoff and Ehrlich. But now in the modern era, we know that, of course, the antibodies comes from B cells, and the protection against infection, infected cells comes from T cells. And again, the helper cells are in the middle here because they can help B cells make antibodies. They can help CD8 cells become cytotoxic cells. And more importantly, I think, they can directly activate phagocytes to kill, engulf and kill microbes. And so helper cells are in the middle of cell and humoral immunity, which I would point out happens simultaneously in all infections. And so I'm going to come back to that point later. So, of course, the other great theory that we have about helper T cells is their specification into into different kinds of helper cell subsets. And of course, the first two to be described by Mossman and Kaufman were Th1 and Th2 cells. And so, of course, Th1 cells are driven by IL-12, and that drives expression of Tbet, and then these cells can become Th1 cells and make gamma interferon and provide immunity to intracellular pathogens, mainly by activating macrophages. And then we've got other flavors of helper cells that activate other kinds of phagocytes. So Th2 cells have their specific program driven by GATA3. They activate eosinophils. Th17 cells driven by IL-6 and TGF-beta, they activate neutrophils. And then, of course, we have T follicular helper cells, which are critical for driving affinity maturation in germinal centers. And so all of these kinds of effector cells can form depending on the innate immune context. And I'm going to talk a lot about type 1 immunity, which involves the generation of Th1 cells. But I'm also going to point out that this type 1 immune responses always drive TFH cells at the same time. And I'll point out that these form by mutually antagonistic circuits. And so that really raises a question of how you get both populations at the same time. And you need the both because, of course, we need both cellular and humoral immunity to clear most infections. And then we have the memory cell theories, probably the most important of those from Keck and Ahmed, where we have memory cell precursors and short-lived effector cells. Those cells die and memory cells live. The CD4 field is much less developed along those lines, although we certainly have the trafficking-based concept of central and effector memory cells. And so we've got central memory cells that recirculate like naive cells through the blood through secondary lymphoid organs. But then we also have effector memory T cells that circulate through non-lymphoid organs via the blood. And then we have resident memory T cells that become resident in various tissues and don't recirculate at all. And so I would argue that we have a lot of very interesting puzzle pieces, but the CD4 T cell field hasn't been great at putting the pieces together to form an intact picture that integrates all these ideas. And so that leaves me with questions like these, do the various effector cell subsets, Th1, Th2, et cetera, have memory cell versions, all of them? And if so, how do they relate to central, effector, and resident memory cells? And then the point I alluded to earlier, how does the system ensure that helper cells are generated to support cell-mediated and humoral immunity in all infections and at the same time? And so I'm going to kind of try to convince you that there are some grand unifying concepts here that can bring a lot of these different ideas together into some kind of a whole, at least a framework where we can start talking about the problem. So before I get into the results, I want to tell you about the experimental system and its core feature, which is peptide MHC2 tetramers. And so the experiments I'm going to describe tonight involve tracking the CD4 T cell response to various kinds of infections. And of course, the important part of the CD4 T cell response to the infection are those cells that have T cell receptors that are specific for MHC2 bound peptides from the microbe of interest. And so the pioneering work of Altman and Davis, showed that you can use fluorochrome labeled peptide MHC tetramers in flow cytometry applications to detect T cells that have complementary T cell receptors that recognize the relevant peptide MHC complex. And so I'm going to describe peptide MHC2 tetramers, where the MHC2 molecule is the IA of B molecule of C57 black 6 mice. So everything I'm going to talk to you tonight, except one experiment, is in mice. In B6 mice, they have one kind of MHC molecule, IA of B. And so we use the the Kaplan and Marc technique of linking immunogenic peptides to the beta chain of the IA B molecule covalently, and then pairing these with IA B alpha chains in insect cells, biotinylate those molecules, decorate streptavidin fluorochrome conjugates, and now we have a miniature antigen presenting cell, 4 identical peptide MHC complexes where the peptide is from a known immunogenic peptide, usually from a microbe. And so I'll talk a lot about this peptide called 2W. It's a minor histocompatibility antigen, or at least a variant of one. And then other peptides, this one from Salmonella, this one from a protein from Listeria. And so in all cases, the red ball here is usually going to be 2W, but sometimes it's these other peptides. And so we use these tetramers in enrichment mode. And we do this because the T cell repertoire is very diverse. And so the frequency within that repertoire of cells that have any particular antigen or peptide MHC specificity is very low. So they're hard to find. And so we realized very early on that unless we could enrich the target, the tetramer binding cells, this was going to be a very difficult challenge, especially because CD4 T cells don't proliferate as much in the primary response as CD8 T cells. So that makes the problem even more difficult. So the way we do the experiment is we take the spleen and lymph node cells or blood, and in one case liver cells from a mouse. We label with the tetramer, and then we add magnetic beads that can coated with an antibody that binds to the fluorochrome part of the tetramer. So we attach a magnetic bead to the tetramer bound cells. And then we take all the cells and we pass them over a magnet, and the tetramer bound cells will stick to the magnet because they also have the magnetic bead stuck to them. And then we can take the magnet or the column off the magnet and to collect the bound fraction, stain with other antibodies to drill down on cells of interest, and then do flow cytometry. And this method was worked out by Jim Moon and Hamlet Chu when they were in my lab. So here's an example of a post enrichment bound fraction of CD4 T cells from a naive B6 mouse. And in this case, we've enriched with 2 different tetramers, both IA B with one with fly C as the peptide and one with 2W as the peptide. And we've had these tetramers are conjugated with different fluorochromes. And so after enrichment from a naive mouse, we have a few tetramer binding cells of each type. They're mutually exclusive, as you'd expect, because they are different epitopes, if you will. So there are different sets of TCRs that will bind them. And they're rare. There's only about 300 2W A of B specific cells in a naive mouse, and only about 20 fly C specific A of B specific cells in a B6 mouse. And they have a naive phenotype. I'll show you that later. So that's pretty, I'm still quite amazed that this technique is sensitive enough to detect the naive cells in the pre-immune repertoire, even before we expose the mouse to the relevant foreign antigens. But if you do that, and so here's a case where we mix the fly C peptide with the 2W peptide and LPS as an adjuvant, and we inject these intravenously and look on day 6 and do the enrichment with these 2 tetramers from the secondary lymphoid organs, we see massive clonal expansion of the 2 populations. And of course, interestingly, the 2W population, the 2W A of B specific population is larger than the fly C I A B specific population, reflecting the difference in the pre-immune repertoire. So we think that's a big reason that some peptides are more immunogenic than others, even ones that bind the same MHC molecule. And that's because they have different representations in the pre-immune repertoire. So we're going to do a lot of experiments with this peptide and this tetramer. So we've studied this in the context of infection by making various transgenic microbes that express the 2W peptide. And we've done a lot of experiments with an attenuated strain, attenuated by Dan Portnoy, of Listeria monocytogenes, a gram-positive enteric bacterium. And this bug's attenuated so that it will cause a transient infection for less than a week, and then is completely cleared from the body. So we think that's important for studying memory because the antigen's gone, but it's a convenient strain, and as I'll show you, it induces a strong CD4 T cell response. So Marion Pepper and Antonio Pagan, when they were in my lab, they looked at the kinetics of the immune response to the LM2W bacteria after IV infection, and then comparing that to uninfected mice after enrichment with the 2W IV tetramer. So again, in a naive animal, there are a few 2W IV tetramer binding cells. They're CD44 low. That's the expectation because CD44 is a marker of antigen experience. These mice have never seen the peptide. But if we get the infection, then we get this huge expansion of 2W IV tetramer binding cells. And that does not occur if we get infect with LM that does not express the 2W peptide. So this is driven by antigen presentation of this peptide as APC take up the bacteria. And Marion and Antonio quantified the number of cells in the secondary lymphoid organs, again, about 300 in the premium repertoire. And then after the infection, massive clonal expansion, producing 200,000 effector cells, about 1,000 times more than the starting number, peaking at about day 6, followed by the classical contraction phase, where 90% of the effector cells die, and then leaving a stable population of memory cells that doesn't change numerically, at least for 2 months after the infection has cleared. So here's the naive level, expansion to effector cells, contraction to bruised memory cells. So we've been very interested in the heterogeneity of the effector and memory cell populations. And so we tried a number of different gating schemes. And so we settled on this one, which was also used by the Carola Vinuesa's lab and Shane Crotty's lab. And it involves the use of 2 markers, CXCR5, which is a marker, a chemokine receptor that marks TFH cells, T follicular helper cells, and PD1, a co-inhibitory receptor. And so at the effector phase, and here we're using the LLO tetramer and just infecting with regular Listeria. You get the same thing with 2W. You get TFH cells that express CXCR5, 2 flavors, germinal center TFHs that have more PD1 and CXCR5 than a TFH population that's probably not in germinal centers, but still expresses CXCR5, but less of it than these and doesn't have much PD1. And then there are CXCR5 negative cells. These cells all express TBAT, I'll show that later, and these are Th1 cells. If we wait till day 60, so the contraction phase has occurred, we now are in the memory phase, and we now have 2 populations instead of 3. We still have CXCR5 positive cells, and we have Th1 cells. The so-called germinal center TFH population is not apparent anymore. And we initially thought they didn't form memory cells, but it turns out that, in fact, some of these cells can become memory cells, but when they do, they downregulate PD1 and CXCR5 and merge into this population. I see someone had their hand up. I just noticed that. I don't know.
I think we're gonna have the Q&A section in the end. So everybody, okay, if you have questions, we can ask in the end.
Okay, thanks, John.
Yeah.
And so, you know, we have 3 effector populations, and then we have these 2 at least phenotypic memory cell populations, or we think this population has inputs from both these kinds of effector cells. And this, you'll see this gating scheme in the literature all the time. And it has some drawbacks, as I'll get to in a minute, but it's pretty useful. And it's led to, I think, a better understanding of the relationship between the Th1s and the TFHs. And so this is the version of an experiment done by Karadi's lab, and Weiss's lab, Chen Dong, Troy Randall. This is our version of it. Everybody gets the same answer with these kind of acute infections. And that is that, and these are done in these mixed bone marrow chimer experiments, but the Th1 cells are highly dependent on the IL-2 receptor. No IL-2 receptor, and you get very poor Th1 differentiation. Has no effect on the TFHs. And of course, the TFHs depend on BCL6. So in a BCL6 knockout, all of the CXCR5 positive cells disappear, and you're left only with Th1 cells. So I'm going to come back to this a lot, but I'm going to refer to this as the bifurcation, where in these kinds of acute infections, CD4 T cells bifurcate to become either TFHs or Th1 cells. And that happens at the same time and produces helper cells for the germinal center and helper cells for macrophages. And so right here we see there's some core feature that is ensuring that there are antibody helpers, or these germinal center helpers, germinal center B cell helpers, and macrophage helpers generated at the same time, ensuring both limbs of immunity. And we think this is a core feature. I'm not going to talk about it much tonight, but for different kinds of infections, like type 2 immunity, Marion Pepper showed you get TFHs and Th2s. For type 3 immunity, at least with acute infections, you get TFHs and Th17 cells. So the bifurcation always happens, but how the non-TFHs differentiate is context dependent. And the bifurcation is heavily influenced by IL-2. IL-2, you get an IL-2 receptor signal, you can become a non-TFH. If you don't, then you can become a TFH, and I'll show that later, or in schematic form later. So this gating scheme has some problems. Right away, of course, it doesn't distinguish memory cells that might come from different effector cell inputs. And we were, based on other work, but from Sue Keck and Rafi Ahmed and Hale and others, there was some heterogeneity in the Th1 population. So we struggled to find a better gating scheme. And so I'm going to show you one possible scheme now that we think is pretty useful. And it also reveals some interesting trafficking features that we weren't appreciating before. And so this is work mainly of Kevin Awesome, a graduate student in the lab with help from Peter Kruger, who's now at Beer. And so here we're doing an LM2W infection. We're looking at an effector time, and we're gating on tetramer binding cells in the secondary lymphoid organs. And so right away, the first gate in this scheme is on cells that express CD62L, L-selectin. And this is important because in the central and effector memory cell paradigm, L-selectin, CD62L, is a classic central memory marker. And so there are cells in the effector phase already that express CD62L, and I'm going to call those central memory precursors. The CD62L negative population can be further subsetted based on a chemokine receptor called CXCR6, which Ciucci and others have shown is a good Th1 marker. So we've got CXCR6 positive Th1 cells, and we have used the TBET reporter, made it in the LI at NIH, to show that these are bona fide Th1 cells. But there's two flavors, ones that express LY6C and ones that don't. And then if we take the CXCR6 negative population, we can display PSGL1 versus FR4, folate receptor 4. P-selectin glycoprotein ligand 1. And this gate probably looks arbitrary to you, where we're focusing on this little nub up here. I'll show you in a minute why we think this is a real population. But if we gate on I'm going to call these germinal center. This is where the germinal center TFHs are, and this is where the regular TFHs are. And that's based on the fact that both these populations express CXCR5. This one in the orange box expresses the most CXCR5. So we think these are the GCTFHs, and we think these are the TFHs. They have CXCR5. Interestingly, these central memory precursors also have CXCR5, and we think these, in fact, are the precursors of both these populations. So that's in the secondary lymphoid organs. If we look in the blood at tetramer binding cells, there are cells in the blood. The central memory precursors are in the blood. Both kinds of Th1s, the Ly6C positives and negatives, are in the blood. The TFHs are in the blood, but these germinal center TFHs are not in the blood. And work from Steve Jameson has shown that they are basically resident in germinal centers. They never leave. So they're not in the blood. And if we look at the liver at this time, the central memory precursors are not in the liver. So even though they go into the blood, they can't get into the liver. They probably don't have the right trafficking molecules. The Ly6C positive and negative Th1s are the dominant cells in the liver, although there's a small population of TFHs, although these could be blood contaminants, and we're working on that now. So if we wait to the memory phase, what we see now, this is post-contraction, early memory. About 90% of the cells are gone, but these are the memory cells that are left. Again, we still have this population, which I would now call central memory cells, because we're in the memory phase. You have LC62L. We've got both flavors of the Th1s still, and we now have what we believe, again, mainly work from Carolyn King and Marco Kunzli, that these are the memory cell progeny of the GCTFH, and these are the memory cell progeny of the regular TFH, based partly on the fact that the pattern looks very much like it did in the effector phase. And you could easily have a model where just every one of these effector cell types has a 10% chance of surviving and becomes a memory cell that's very similar to its effector cell, but is quiescent. And so we kind of like that model, and I'll show you a test of it in a minute. Here again shows the downregulation of CXCR5. The CXCR5 positive subsets, this one, and this one, still have some CXCR5, but a lot less than they did as effector cells. Again, that makes CXCR5 a lot less useful as a marker, we think, of TFH-derived memory cells. So we like this gating scheme better for that reason. Now, if we look in the blood, again, the central memory cells are in the blood, as you'd expect. The Ly6C positive Th1s are very abundant in the blood, but the Ly6C negative Th1s, which were in the blood at the effector phase, are now missing from the blood. When we think that's consistent with the Ly6C negative Th1 effector cells go into tissues and never leave. They become resident. And we have a parabiosis experiment now that will test that directly. And again, there are, these TFH cells are also recirculating in the blood. And then the liver looks a lot like it did at the effector time and dominated by both kinds of Th1s, Ly6C positive and Ly6C negative. And so the fact that the Ly6C negative Th1s are in the liver, but they're not in the blood, but were in the blood, again, is consistent with they migrated to the liver and became resident there. So we think these are basically these are recirculating Th1s, and these are resident Th1s. So we wanted to test that idea that effector cells just turn into similar memory cells when the antigen disappears. Maybe this is obvious, but I felt like it really hadn't been tested well. So Kevin did the experiment where infected with LM2W, waited 7 days, and then sorted the various kinds of effector cells, the TCM precursors, the 2 kinds of Th1s, and the TFHs. We didn't sort the GC-TFHs because we know they don't traffic through the blood, and we were going to inject these into the blood. So we thought that was not a reasonable thing to do. But anyway, we could figure out what happens to these cells. So we put them into an infection-matched mouse at a time when the infection is waned dramatically. There might be a little bit of antigen left, but not much. And then waited 21 days for the cells to transition into the memory phase, and then asked what subsets do they make. And so the take home is they basically make themselves, and that's pretty clear. So if we put in LY6C negative Th1 cells and then wait and look at what memory cells come from that donor population, they're mainly LY6C negative Th1s. LY6C positive Th1s, kind of the same result, although they definitely generate some negatives. LY6C negative Th1s, and the TFHs really, as they go through the contraction, generate themselves. The central memory precursors, interestingly, they're the only population that generate memory cells of that kind, but they also generate a lot of TFH cells. And we think that's because there's a little bit of antigen, probably that drives them to the next phase in their differentiation. So the bottom line is we like this kind of freeze in place model where the antigen clears and then everybody just becomes a quiescent effector cell of the type they were before. So I just want to point out that this bifurcation also happens in humans. And so we got an opportunity, Sean Mahmood in my lab, to work with a group who was studying the yellow fever vaccine and was able to sample lymph nodes by surgical resection, believe it or not. And so we had made tetramers that contained peptides from the yellow fever virus envelope protein bound to either DR11 or DR15. And then we had people who expressed either DR11 or DR15. And we got 4 million lymph node cells. So many more. So in our mouse experiments where we do enrichments, that's 200 million cells of SLO. Here we have lymph node cells, 50 times fewer, but they're from the lymph nodes of a human. And so if you're following what I just said about the trafficking, we can't study germinal center TFH cells in blood because they're never in the blood. So here was a chance to look at this. So we got an inguinal lymph node from these people on one side before their vaccination, and then an inguinal lymph node on their other side after the vaccination in their thigh, so the draining lymph node. And so you can see there are very few tetramer binding cells in the pre-vaccination sample, but we can find a nice tetramer binding population after the vaccination in the lymph node. And most of the cells are CD45 RA negative, as you'd expect for antigen-experienced cells. And if we phenotype these cells, and in human immunology, they like to use chemokine receptors for everything, based on the work of Federica Salusto. And so a Th1 associated chemokine receptor in humans, in fact, and in mice, is CXCR3. And so what you can see is that if you display the tetrabinding cells, CXCR3 versus CXCR5, we have Th1s and we have TFHs, and they all express CXCR3. So maybe we should call them TFH1s. This is also true in the mouse, actually. In our LM experiments, the TFHs in the LM infection also express CXCR3. So not and we looked at now 5 people, and it's about 50-50 on average in between Th1 and TFH1 in the lymph nodes after the vaccination. Now, there are other memory cells in the bulk CD45 RA negative population in these people that we don't find in the the yellow fever specific population. And I should point out the vaccine is an attenuated virus. So we don't see TFHs, that are not TFH1s, but you find those in the bulk. And then there's a bunch of other kinds of effector cells, which we think are Th2s and Th17s that don't appear in the virus infection. So it looks like a mouse. So that made me happy. So this is the model that we prefer. So naive cells during infections that see their cognate peptide MHC2 on an activated APC, they get signal 1, they get signal 2, and they start proliferating. And then some of them are able to bind to IL-2 and some aren't. And we can talk about where that heterogeneity comes from at the end maybe. But the cells that get an IL-2 receptor signal turn on STAT5 and BLIMP1. BLIMP1 is a repressor of the TFH fate. So these cells become non-TFHs. And because this is a type 1 immune response, these cells differentiate, these non-TFHs become Th1 cells. And actually, maybe I'll get to this. We think this early Th1 differentiation is actually independent of IL-12 receptor and depends on IFNAR, at least in part. If cells don't get an IL-2 receptor signal, they do not turn on BLIMP1 and they turn on BCL6. And then if they get additional STAT3 signals from IL-6 or IL-21 receptor by binding those cytokines, they'll become a TFH. If they get an ICOS signal, they become a GCTFH. And many of these steps come from work of many different laboratories, but with different kinds of acute viral and bacterial infections. And we think that there are some cells that do not get IL-6 or IL-21 signals, and they're kind of stranded in this more naive-like state. These are the pre-central memory cells. And then we think when the infection clears, all these cells, the 2 different kinds of Th1s, the 2 different kinds of TFHs, and the pre-TCM, all form memory cells. And they have different recirculation patterns resembling, effector or central memory cells. And so, of course, these cells appear to be recirculating through SLO. So these would be central memory-like. We think these cells are resident in the SLO, so they're like TRMs in some way. These cells are recirculating through SLO and non-lymphoid tissue. So these are like effector memory cells. And these cells are resident in SLO and NLT, and again, we think are a form of TRM, although we need to prove that or test that formally with parabiosis. And as I mentioned, we think this bifurcation happens in all infections. If it's a type 2 immune response, TFH and the non-TFHs become Th2s. In a type 3 immune response, TFHs and the non-TFHs become Th17s because there are different cytokines present. So, after working on Listeria for many years, I got this email from Ian Orme, who's a famous immunologist working on TB infection. I guess I received this on June 1st, 2016. Ian says, "Hi, Mark, catching up on my reading. Immunity to listeriosis is mediated by CD8 cells. So why do you study CD4s? Am I missing something here?" And so for me, that was kind of like a Simpsons moment, like, don't. And I got very worried.
Ikke ejeren bilen her bag mig næste gang overvejer shit.
I was worried that we were really, doing some what I thought was sophisticated cellular immunology on an infection where CD4 cells weren't that critical for immunity. So what infections are CD4s actually critical for immunity. And we know this from people who, of course, have HIV and have all their CD4 cells depleted by the virus. And there are people who are born without CD4 cells. And there are people who are born with deficiencies in the IL-12 receptor and other aspects of type 1 Th1 differentiation. And people with these deficiencies get these kinds of infections. TB, Salmonella, Toxoplasma, Leishmania, various kinds of yeasts that can cause problems. These all kill AIDS patients who are not treated. Conspicuously absent in causing more severe infections in AIDS patients are influenza virus, streptococci, and worms. So that I still find this quite odd. But I felt like, gee, we've been studying immune response to Listeria, which I'm going to show you are very similar to what happens with flu. And yet the helper cells aren't that critical for the immunity. We should be studying one of these bugs. So I'm going to show you, and so we picked Salmonella. And I'm going to show you a comparison of the response to these two different kinds of bugs. And which leads to an amendment of that bifurcation model that I just showed you. So we made our collaborator Ryan Langlois made an influenza A virus that expresses a 2W peptide, and of course this is an RNA virus, segmented RNA virus, strong type 1 interferon inducer, and it gets cleared from the body. And CD4 cells, depends on whose paper you read, aren't that critical for the immunity, and certainly it's not a major opportunistic infection in AIDS patients. So Salmonella is a gram-negative enteric pathogen. It causes a persistent infection of macrophage phagocytes, which it has in common with all the other pathogens I showed you on the previous slide. They're intracellular infections of macrophages. And we know from work of many investigators that these infections are controlled by CD4 T cells, but not eliminated. So they keep the infection at bay, prevent the infection usually from killing the host, but you don't get sterilizing immunity. And so my former postdoc, Stephen McSorley, made a recombinant 2W strain expressing 2W. And then Peter studied the immune response to these infections. And so he just tracked the number of cells for either Salmonella 2W or IAV 2W. Here's tetramer binding 2W cells. So for Salmonella, we get massive expansion, peaks at almost 1 million effector cells, and then contracts and goes into memory. But again, the infection doesn't clear. So it's highest here, but then falls to a very low level, and we think is controlled by these T cells. Now for flu, you start at again about 300 naive cells. There's also clonal expansion. It's lower, it peaks lower than for Salmonella, and it's delayed a bit. So by day 4, where you're already getting clonal expansion for Salmonella, we're not getting that for flu, but definitely big time clonal expansion in both cases. So we phenotype the cells, and I'm going to show a little different kind of phenotyping here, but it's kind of like that old-fashioned phenotyping where we're using CXCR5 as a major phenotyper. So we're displaying BCL6, the lineage-defining transcription factor for TFHs, versus CXCR5 in our first gate. So if we look at the naive 2W binding population, they don't have BCL6, they don't have CXCR5, they don't have TBAT, they don't have viral T, they don't have CXCR3, and they don't have CX3CR1, two interesting chemokine receptors, as I'll show in a minute. They're naive cells, no surprises. So if we look at flu, again, here's all the tetrabinding cells, day 4, day 7, day 10, day 50. And we see that pattern. It looks exactly like we saw for attenuated LM. We see, at day 4, the response isn't really getting going, so they're mainly naive. But now by day 7, massive clonal expansion. We're at this point here. we've got TFHs, GC TFHs, regular TFHs. We gate on these cells, they all have TBET and they all have CXCR3. So we've got two kinds of TFHs and we've got TH1s, just like we did with LM. And then as we progress into the memory phase, we start to lose this population. It morphs into this population, and we're left at day 50 with a memory population of, we now know, inputs from the central memory precursors from GCTFH and from TFH in all likelihood, and then from TH1s. And the TH1s have TBET and CXCR3, not CX3CR1, and there's no TH17s that form in this infection. So it looks a lot like attenuated LM. Experiments done by Sarah Teichmann, Hake, and others on acute malaria infection show a very similar pattern to this. So lots of infections showing the bifurcation and the formation of the two kinds of memory cells or sets. And I'm going to call this module 1, which is the bifurcation in type 1 immunity. So we look at Salmonella. If you look at day 4, where we're starting to get clonal expansion, it looks a lot like the bifurcation. So we've got we've got TFHs, germinal centerkine, non-germinal centerkine. We've got TH1s. They have CXCR3, but they don't have CX3CR1. Pretty much looks the same at day 7, although the TFHs seem to be going down. Maybe a hint that this CX3CR1 population is starting to appear. And this binds fractalkine, this binds two other chemokines, CCL9 and 10. But by day 10, where we're at this point in clonal expansion, where we've had this massive increase beyond what you get with flu, now suddenly we have very few TFHs, at least as a percentage of all the cells that are there. The TH1s have a little more TBET that they did at day 7, or that you find in flu, and now you get the appearance of the CX3CR1 subset. And by day 50, hard to find any TFHs, and you have this almost 50-50 split between these two kinds of TH1s. Now, I'll point out, if you look at the absolute number of TFHs, so that's looking at all the CXCR5 positive cells, that's this curve compared to flu, where we see this curve, the open triangles. So the absolute number of TFHs isn't that different in the two infections. What's different is that after a week, the TH1s keep proliferating in Salmonella. And as they keep proliferating, they start diverging into these two subsets. So I'm going to call this an amplification model. So module 2, where you amplify the TH1 limb of the bifurcated population. And I would point out that if we look at Salmonella 2W at day 10, and we look at various knockouts, the IL-12 knockout, at least the P40 chain of IL-12, that reduces that outgrowth of TH1s, or at least reduces the cell number. That's not true for the IFNAR knockout or the IL-18 knockout. And if we look at the cells that form in the IL-12 knockout here, and of course the control is the wild type, where this module 2 amplification has occurred. Basically all the tetramer binding cells here are TH1 cells. But if you block IL-12, you reduce the number of cells, you block the outgrowth, and now you see the reappearance. At least the TFHs are now back to a larger fraction of the total population. Now, IL-12 deficiency does not completely prevent TH1 formation. You still get TH1s. And this looks a lot like what we see with flu, a lot like what we see with attenuated LM. So there is an IL-12 independent path to TH1s, And that happens with these acute infections, or at least the early part of the bifurcation. But if you need IL-12 to amplify the Th1s so that they outgrow the TFHs and then start to form this population of CX3-CR1 positive cells. And so again, the IL-12 knockout, the percentage of Th1s goes down, but not to zero, they're still there. And that CX3-CR1 population it goes away. So that's our IL-12 dependent steps. And I'll just quickly, the P40 chain of IL-12 is shared with IL-23, but we've done the experiment in the IL-12 receptor beta-2 knockout, which is an IL-12 receptor, IL-12 specific receptor. And we see the same phenomena where we see the reappearance of the TFHs as a large fraction of the population and loss of the CX3-CR1 positive cells. So that's IL-12, not IL-23. You can do the same experiment by blocking with an antibody. So here we've done the infection, we neutralized IL-12 with an antibody. And again, when you do that, you block the Th1 outgrowth, you have TFHs as a larger fraction of the population, it's quantified here. And actually, if you purify these two kinds of cells, transfer them into naive mice, and then challenge with salmonella, this population protects the liver or the spleen and the liver a lot better than this population, which is a mixture of these module 1 Th1s and TFHs. So these module 2 Th1s are really important for protective immunity. So I'll end here. This is the model I showed you before. And we think this happens, with all these type 1 immune responses. But in salmonella, an infection that persists at a low level, Dan Barber's reported very similar results for TB. These Th1 cells that you get from module 1 get amplified dramatically by IL-12. They proliferate like crazy, outgrow all the CXCR5 positive populations, and then differentiate further to cells that I didn't show this, but make more gamma interferon than these kinds of Th1s. And then we think sequentially differentiate based on a transcription factor called ZEB2 into this population, which, we're not sure what these, which of these cells is the more protective. We think it's these actually, but these cells might be in different places in the body and do different things. And so really, there's 4 kinds of Th1s, these 2 from module 1 and these 2 from module 2. And so what are their differences? What are their functions? I think that's research for the future. So I'm going to stop there and take your questions, and thank you for your attention, and forgive me if I've confused you.